Okay, I think we can officially start. First of all, thank you all for registering or not registering and coming here. It's a really great honor for us to see that you're interested in the hyperspectral imaging as it is, processing in our work in general. And probably the first thing I would like to ask you is technical moment. We don't have permission to use all the power sockets which we have. We have quite a lot in the floor. You see those holes. It's not permitted because of the technical reasons, safety reasons, yes. So first 20 minutes of the workshop will be the presentation. I will go through the introductory information. So if you don't want to stay without juice for the laptops, maybe it's makes sense to save the battery for the second half of the workshop. Okay, and yeah, after the introductory part, we can also go for a short troubleshooting session, like 10-15 minutes, for those who tried to install the environment for themselves, to try to do the stuff of their own. If it wouldn't be necessary, great, but we will try to just address all the errors if questions will be present. So let's start. And as I said, welcome to our workshop. Firstly, I'd like to introduce you. Oh, it's work? Yeah, I think it's working beautiful. So first of all, I'd like to introduce our company. And Photon Systems Instruments, we are located in the Czech Republic, in the Brno County, quite far from the main city, from Prague. We are producing quite a lot of equipment for plant phenotyping, starting with the smallest handheld devices, which you can see in our booth. So nearby in the hall, all the way through the portative laboratory devices like the FluoroCam, like compact one, which you also can find in our booth, and going to the all-in-one modular systems where you can find automatic plant or sensor delivery to the object, to the plant object. So where you have the controlled conditions, where you have set of 5, sometimes 6 sensors, we have our research centrum nearby, in which we are developing new devices, testing new devices. We are conducting experiments in partnership with our friends from all across the globe. So if you have a chance, if you have interest, you can drop us a line and we'll be super happy to show you everything what we are working on, because what you see on our booth, what you see in our presentation is essentially only like the small visible part of what we're doing. And if you're interested in some prototyping, if you have some ideas, if you think how we can adapt to your research, you can come to us, you can communicate with us, and we can just definitely imagine something together. So, and now let's go for the, okay, 1, 2, 3 seats, and 4, why not, if needed. So yeah, let's go for the introduction into the matter. The workshop core and the subject was the hyperspectral imaging, and like any imaging, essentially, which is taken in the field, natural conditions, it works with the reflected light, which is called reflectance. So there are some sensors, usually handhelds, which are working with absorbance, so the light which goes through the leaf. We are working with the reflected light. So reflected light is everything that you see around. Usually in the hyperspectral or any spectral really measurement imaging, we are working with the quite defined chunk of the spectrum, because light spectrum, as you know, goes for millions and millions of nanometers. So we are talking about the visible part of spectrum, so-called vis light. So starting from ultraviolet, ending at 400 nanometers and finishing at roughly 700 nanometers. Then near infrared, this is a range which we don't see, but which is of critical importance for the evaluation of the vegetation, plants, health, and so on. And usually, considering that they are close by, they're united in so-called near range, so visible plus near infrared. And we also measure in the second part of the spectrum. So this is shortwave infrared. It's starting from the roughly 900, 1000 nanometers and ending somewhere at 2500 nanometers. Usually it's used for the evaluation of the water stress, nitrogen content. Sometimes it's used for cellulose content, or from my experience, it was not very successful, those tries which I saw, but okay. And the visible near range is usually used for the estimation of the general plant fitness because it's evaluating like visible light. So we see it's green, red, or stressing yellow. So it can see the very fine differences in the living plant spectrum. And this information is usually measured by three type of carriers. Those are satellites or drones, which are conducting measurements from high above. So in this case, we're talking about so-called remote sensing. In case of satellites, its resolution now is quite good, so you can monitor your field, but it's still the level of rather the plot of plants, so it's like several meters resolution. In the case of drones, it can be separate plants. Now drones are quite good, and I've seen the images where you can really catch the separate plants, and you can distinguish that those are separate plants. We are working as Photon Systems Instruments company. We're mostly working with the so-called proximal sensing, so we're getting closer to the plant, so we can distinguish not only separate plants, but separate organs of the plants. So we're getting closer to individual plants, which is very useful for study, not only for the monitoring for the smart agriculture, but essentially for the development of new agricultural techniques. So what I mean by the proximal sensing is that camera is located on the crane or robotic arm, which is moving like a meter or two above the plant sample. And visual resolution is quite fine. So all of those sensors are producing not just images like RGB or black and white, but it's producing so-called hypercubes. And those hypercubes can be, so they are roughly represented here, and they be kind of spooky stuff because it's not so easy to figure out what's it exactly. The hypercube is essentially a 2D image, which is taken, so the spatial dimensions, width and height, which are taken at every nanometer or every several nanometers of the scan spectrum. So we are going through one of those chunks and take an image at every nanometer in our case. And you can see that if you take one particular pixel, like it's shown here, you're getting some value of this spectrum reflectance. And if you're going through all the spectral axis, which is here represented by this z-axis, actually in this image is rather y, but it's marked like z. So if you take one particular spectrum of a particular pixel, you'll get the whole spectrum, so like it's written here. For making it a little bit clearer, RGB image would be essentially the same stuff, but it will have only 3 of those images, only 3 of those frames, and it will be only 3 values in this, so for red, for green, and for blue. So essentially, RGB image is the same. RGB camera is also spectral camera. Simply, the resolution is not very good, so you cover very big chunks of spectrum. So we are doing the same kind of imaging, but with much finer resolution. And actually, one more intuitive stuff which helped me a lot to wrap your hands around the hypercube, because when you imagine that one pixel is a spectrum, it's fine. You can think about this, you can imagine this, but when you try to imagine that every pixel is a separate spectrum curve, your brain can burst, because it's not very intuitive for us to operate in this kind of 3-dimensional space. We are used to proper 3 dimensions where all the dimensions like height, width, and depth are spatial, so they're the same quality. And here you have kind of quasi 3D. You have spatial dimensions, height and width, and the third dimension is spectral. It's something which you cannot touch. And for my intuition, it helped a lot to imagine hyperspectral cube like video. Because in case of video, you have the same quasi 3D structure. You have width of the image, height of the image, and you have depth, which is covered by time. So if you imagine the hypercube like a kind of video, and you walk through the spectral dimension, it starts to be a little bit more intuitive. You start to see what exactly is happening with the plant and with the background, where you're going through the spectrum. So it's so-called spectral walk. So see here, you see the range at which we're going. So here you see the plant spectrum curve, and you see how it changes through the spectrum. So now we are getting into green area. Plants are reflecting most of green light, so we are getting some rise in the signal. Now we're getting deep in orange and red, because plants are usually not orange and red. And now we're getting to far red, which I mentioned that it's super critical for the plant health evaluation, because here we have so-called red edge where the signal just skyrockets, and we can, by the intensity of the signal, evaluate quite a lot of information about the plant health. So we will be quick here. It's intimidating, I know. So, but this is essentially just very brief introduction to how those hyperspectral cameras are working. So we have 2 main types which are used in our cameras. So this is line scan and the wavelength scan. So the problem of taking hyperspectral camera, hyperspectral images is in the fact that sensor is 2-dimensional and you need to capture 3-dimensional image. So you need to sacrifice 1 dimension. If you are doing wavelength scan, you are taking XY dimensions, so height and width, and then you swap the filter. So you're going across the spectral axis. If you are doing it with so-called push broom or line scan, you're taking width and spectral dimension, and you're sacrifice X dimension. So you are getting rid of 1 X dimension, and then you're kind of capturing video and scanning your sample only 1 row of pixel in the width, so 1 row of X pixels. You are going forward and you're measuring all the spectral information behind. So, and essentially the push broom, so this is main method how we're capturing our hyperspectral images. Push broom method is essentially concatenate together video, where you take line by line of the hyperspectral image pixels. So the second one, this wavelength scan, is what we use in multispectral camera, because you need either filter wheel, which will be cutting chunks of the spectrum, or you need a set of filters like mounted together. And in first case, you're sacrificing a lot of time. In the second case, you're losing a lot of signal. So amount of those filters will be quite short. So that's why it's good for multispec, but not very good for hyperspec, where you have a lot of wavelength. So, and our cameras are mounted into different carriers, as I mentioned. So the classic one, lab one, is the plant screen system, where we have a constant light source when we have our camera mounted in the robotic hand, which is automatically adjust the height, and which is scanning our plants in this direction. So we are illuminating light, and we are scanning like this, taking row by row by row by row every pixel of the image. And the second possible carrier is so-called fill screen system. So we can mount our cameras into the tractor. And in this case, stuff is getting a little bit trickier, but we will go to this when we start the workshop, because tractor is a device which is unable to move so smooth the robotic arm, and the illumination conditions in the field are particularly challenging stuff to solve. We'll go to this because we will be working today mostly with the field data. So, and when we've got our hypercubes, where we got our spectrum, we are getting a problem. maybe rather unexpected ones, but still, when you have not 3 channels like in RGB or not 6 channels like in the multispectral imaging, and you have 400 nanometers, 400 images, 400 data points for every image of your data, the critical question is which are meaningful and which are less meaningful. Because spectra in the visible plus sphere range of spectrum, they're very smooth. So it's not like in the mass spectrometry or the Raman spectroscopy. So you don't have dedicated peaks, which you say, so this peak position height is responsible for this particular chemical. We can just evaluate this one and get rid of anything else. Our peaks are smooth. So our importance, our variance, if you want, is distributed evenly through the spectrum. So you have heavily dependent data, so they're not independent. And you need to go those sometimes not particular wavelengths, but also chunks, which are important and which is not very easy because your plant is not stable. It's not like stable thing. You have different illumination, you have different vegetation conditions. And so for this, we need to figure out what's important. And the easiest way is so-called vegetation indexes, which are like useful mathematical shortcuts. Those are a combination of 2 or 4, 3 bands, usually not more, combination together, and a lot of those published, and you can just talk those and easily calculate some traits. For example, NDVI, so this one, which is 800 nanometers and 670 nanometers combined, is responsible for the general health of the plant. So you can evaluate how green the plant is. So for example, on this heat map, you can easily figure out which plant is not healthy and which one is a little bit in better shape. And for example, this anthocyanin reflected index is kind of useful for estimation of anthocyanin content. So you see the ripe strawberries are much more have the heat map, which is much more saturated, bright in case of the ripe strawberries. That is not reacting anymore. Okay. Oh, we're stuck. Okay. So then the second, don't be afraid, the second part, how you can evaluate your result is non-supervised part. So before this, this vegetation indexes, which are good, but not always fit to your particular research because you found them in the literature, and literature is rigid. I mean, you can try to adapt it for your particular case, but sometimes your case is so different that you simply don't have any vegetation index which fits to your needs. Sometimes, and in this case, you can kind of find flexibly in a particular case what threads, what wavelength, what region of the spectrum is important with techniques like principal component analysis, which is used for feature extraction. Feature extraction means that we're not taking wavelength, but we are rather composing new features, which are like combining more of wavelength or original features inside. To make you a very brief understanding what actually principal component analysis is doing, it's shrinking dimensions, and here one example. So our spectrum has 400 wavelength. Let's drop it to 2. So we have wavelength 1 on the axis X and wavelength 2 at the axis Y. If we plot them, we'll get a scatter plot. And we are not happy with working with the 2 dimensions. We want to shrink it to 1 dimension. And PCA is projecting those 2 wavelengths to 1, where it finds the most information stored. So which position it will be? So here is mark. So most of variance is remained, most of the spread of the data is stored, so we can sacrifice rest of information to get less dimensions. Principal component analysis can be used for also defining what wavelengths were the most important for those new principal component generation, and we will cover it in the practical part as well. So here will be just 2 short examples how the spectrum information can be used for the real agriculture insight. So here it was a study which was done on our plant screen modeler system, which was people were doing absolutely crazy thing, which I like very much. They were evaluating the whole spectrum, so they were putting the whole spectrum into machine learning model and evaluating the correlation between the particular wavelength regions of the spectrum with the biological threats. Crazy complicated, very interesting, super informative. So the second thing, which I found quite funny, because people here were evaluating the shadowing of the hyperspectral images, and we kind of bumped into the same problem independently in our system. So here they were evaluating the spectrum information to segment plants in the field and drop those values which were shadowed, so skewed. That was very interesting because you can do it also very efficiently. And those studies are like very many, but mostly they're located in the remote sensing, so it's drones or satellites, and not so many are in proximal sensing. So we are getting close to the plants. This is not very much open area for now, so we are trying to make it more open, more used. So, and the last part of introductory, it will be the short introduction to the case study with our partners from Katrine Institute in Palatsky University in Lomouc. So we together were going to the field and evaluating the wake up. What's going on? Yes. So I'm evaluating the calendula cultivars, trying to extract useful information from this. We're using field system for this. So hyperspectral camera mounted into the tractor. We were evaluating 10 cultivars of calendula. And we were trying to, first of all, figure out what method will be best for the evaluation of their phenotypic traits. We will try different sensors. Then we tried to create the pipeline which will be best for processing of those fields data, because field is crazy complicated. You have constantly changing sun conditions. You have plants which are varying very much more than in the lab. You have essentially the biggest problem is the illumination conditions, because you have different cloudiness, you have different position of the sun, the spectrum is changing all the time. And the last thing was to compare different machine learning models for evaluation of such data. So gather data, process them, and then get it work for some insight we can get from it with machine learning. So we were measuring it with the field screen system, polypen spectrometer, RGB camera, field screen system, which was taking the hyperspectral imaging, was by far the most efficient one. I hope soon it will be a publication about the comparison of those and why the hyperspectral imaging was superior in this case. So yeah, that's how it looked. So we had a tractor with mounted hyperspectral camera with the shadowing curtain, which was homogenizing the light, at least to the degree it's possible. As you will see further, it's not always possible. And we had the camera. So, and here's the result. So essentially now I'm starting to go through the slowly through the practical part of the workshop, because we will be doing in code exactly this when we just move to the code. So a little bit of background information what's going on. So here we have the result of the hyperspectral scan. It's raw hypercube. The resolution here is tricky because it's very good in the spectral and in the width dimension, but not very good in the y dimension, because as I mentioned, tractor is thing which cannot move evenly and which moves a little bit too fast for gathering our kind of data. So in case of y dimension, it's not blocky like the old blocky. So we have in X, we have quite reasonable resolutions, 600 or 700 pixels. But in case of X direction, y direction, it's rather lower, and we had to work it around, because it's kind of one of the standard limitation of the field imaging. You always have something like this. Anyway, so in this image, you see the raw hypercube. You see the white balance, or the combination of the channels, is completely off. You see that we have heavily difference between the illuminated flowers and shadow flowers. You see huge difference in the reflectance. And overall, you see that probably during this measurement, the sun was somewhere under the cloud, because even the white reference, which is supposed to be white, is just dark and almost invisible. So if you look at the spectrum of the illuminated plant here and the shaded one, you see the difference is immense. And the shape of the curve itself is weird. It doesn't resemble how reflectance of the vegetation should look. So what we've done, we done so called wife reference step-on collaboration. So we measured the homogeneous spectrum of the reference. You see that the spectrum is itself quite skewed. So, and when you apply transformation to compensate for this like default curve, you're finally getting your normal vegetation shape. And the second part, it was white reference on calibration. This thing is critical for the field because usually this part we are doing also in the lab, but this part we are doing only in the field. And the necessity of this is one of the reasons you can see here that the spectrum of the sunlight or the noon and somewhere just later, it's not only like higher or lower, the shape is changing. You need to compensate for this kind of shape changes. You need to compensate for the different amount of humidity in the air and cloudiness, and for this you need not only measure the before the experiment, the wife reference, but you need to have the reference in every image of yours. And for this we had the white reference in every frame. It was mounted under the camera, and we're also compensating for this. So after those spectral compensation, you see that wave balance is already fine. So we already have normal spectral shape. We're already looking at something which at least by color resembles the calendula. So, and then we did several steps additional, which we will be doing, in the programming part, which are hidden here. And the next part was actually compensating for the shadow. Because as you can see, some flowers are just cute and bright and some are hidden completely. And here we used one of those normalization methods which are available in our library as well. And after, I think in this particular case, for the visualization purposes, I was using normalization by a particular wavelength. So every wavelength was divided by, I think, 750 nanometers. So it was like local peak, so essentially maximum normalization. And after this, when every pixel of the image was normalized in this way, you see that we are essentially getting rid of most of the difference between the shaded and the illuminated spectra, though not completely. And this is important, and this is actually okay. So, and then logical step is after normalization, so preprocessing per se is over, and we need to extract our pixels, plant pixels. For this, you can use manual annotation. And in our case, we were using, we were collapsing our hypercubes into 3 channels. So first channel was photochemistry. I don't remember the okay, so it was PRI, sorry, the exact deciphering of the name. It was used for the segmenting of the flower pixels. Then we used NDVI for filtering out the green vegetation pixel because we don't need it. And then we were using blue NDVI, so it shifted into the blue range of spectrum because it was surprisingly efficient in the segmenting soil out. So sometimes soil was also interfering into the our pixels. So essentially, then we use machine learning model to find ideal weight for each frame. And we did it automatically with manual annotation. Here now we will be doing it a little bit different because setting interaction annotation was rather tedious. We can speak about this some different time. And so here in this workshop we'll be doing manually thresholding adjusting. And then we got like 3 separate masks for every index. And when you combine them, you are getting the ready plant mask. This is rather shortcut. So this thing can be made much more efficient and sophisticated, but it was enough because we have a lot of spectral information. We're good with such shortcuts. And then we scanned over many machine learning models to figure out which one would be the most efficient one for the, in our case, classification between different classes. So we wanted to create the detector which will be able to detect, classify efficiently every plant cultivar from each other. Because like if you plot all the spectra, you can see that they are very similar. And if you are able to do this classification task, you can go for the next level. You can go for the regression task and predict particular chemical compounds. So with our tests, across all the classical machine learning methods, like we found like two favorites of mine. It was random forest plus principal component analysis, support vector machines, and if we go a little bit deeper, we can get more efficient, and we were quite happy with one-dimensional convolutional network. In our workshop, we'll start simple. We'll start with something linear. So we'll go for logistic regression because it's fast, quick, and easy to understand. And if you're interested, if we'll get some feedback, we can show you in one of the new workshops how you can scan and tune and find the model which fits you. So as a result, we got the quite efficient classifier. As you can see, the metrics here are very close to 1. So that means that almost all the time we're getting our class right. So I recall, I'm saying again that it's 10 class classifier, so it's quite good, I would say. And the good thing of one-dimensional CNN is that it's actually it's like tape convolution network, but still quite shallow, so it's very nicely interpretable. And we can plot our embedding maps and get some insight from there. And you can see that though shallow, we still can get some useful features from there. You see that most of the population of our cultivars are separated into 2 groups, so shadowed, as I mentioned, pixels, and not shadowed, but they're still kind of united together. So our model was not we didn't intend this, but was able also to classify shadowed and illuminated pixels even after the post-processing. So good. And then you can show which wavelengths were critical for this as well. You can cluster those cultivars, and yeah. So essentially, I'll introduce this vision of what should be done next, and we'll move to the practical part. So what should be done next is we plan to combine all the sensors we were used. We already have quite extensive results from the mass spectrometry, so we have biochemistry in hands, and we plan to use all the sensors of information to create smaller models and then to load them into one big meta-regression model to use completely non-destructive pipeline for evaluation of the biochemistry compound in calendula. So this is our big plan. We're working on it, and now, just again, don't work. Yeah, so thank you. I would like to thank our partners, and we are moving to the question part and to the practical part. So if any questions to the introduction are here, I would be happy to answer. Hello. In the second part of the workshop, which will be connected to the first part, we decided to separate more detailed information about the cloud environment deployment, so how you can get access to the Colab notebooks, and in more slower pace go step by step in the processing pipeline. So in the first turn, in the first step, we will follow instruction from the mail, which every participant gets from the workshop, and will install Colab environment for the notebook execution. So I'm finding the original mail. To the participants, we can use either the link to the OneDrive folder or the link to the Google Drive folder. It should be more or less the same size, around 150 megabytes. So here we copy the address and by pasting it into the browser of your choice. So here is our folder. We can go one folder up, select it, and then load. It might take some time, depending on the speed of your internet connection, but essentially it should be quite quick because, as I mentioned, the size of the folder is around 150 megabytes. On the other hand, as I mentioned before, you will need a Google Drive account, a Google account, which you can just register only for this workshop if you don't want to have it long term. Google is quite okay with you supplying it only basic information for the dummy account, so you even don't need to set your real name. And it will provide you with 15 gigabytes of storage, which is exactly sufficient for us. So as you can see, the download finished. I was checking before, so these are two identical files. We can show it in the folder. Yeah, here it is. We will unpack it in the folder. Here's the window. We'll extract all the content inside. Here's our folder, and we are copying it, or we can just drag it into the Google Drive. So in Google Drive, you're going to the folder My Drive. And here we have to create one additional folder, Colab notebooks. Here it is. So now we are pasting our workshop content inside. So you see the program here, progress. It should be also quite quick. Okay, so all files are downloaded. We can check it with the entering the folder. The path to the folder should be as following. So My Drive, Colab Notebooks, 2025 EPPS, and then we can actually already launch our workshop. So here we'll click right button, open with, and here we don't see Google Collaboratory. That means that we need to install one. So we're clicking connect more up. And here, instead of scrolling, we are just writing Collaboratory. See, it knows what we're looking for. So, and just install. Yes, we want to continue. It asks us for the permission to connect to the Google Drive. Considering that Google Collaboratory is a service of Google, it is quite safe. Letting it connect. And after a few seconds, everything being installed. And we are done. We're closing this menu, and now when we click on the right button, we see, okay, probably we should just start. Yeah. We had to close and open the browser, but now it hopefully works. So now we can open it with Google Collaboratory. Just right click, open with Google Collaboratory, and we are in. Now we can finally go to the workshop content. So here you have intro slide. Then there are several sections. First of these sections is description of the workshop, which you can refresh in your mind. Essentially here it describes what the library we are studying here is used for. Its name, HCI-PSI, Hyperspectral Imaging from Photon Systems Instruments company. The main purpose of the library will be shown afterwards. So then there is a section with the general structure of the notebook where every point is described. Section -1 is introduction, where the library itself is described, what modules it's made of, and what files it's working with. Then we are describing shortly and conducting workshop, so workspace preparation,
we load and install necessary libraries and mount in our Google Drive storage. So then we are moving to the actual processing. When on the first step, on the first several steps, we are conducting the preprocessing pipeline on a single hyperspectral image, moving step by step from loading, then to sensor calibration, spectral despiking, solver correction, spectral smoothing, then range cropping, and as a final step, normalization. So essentially, preprocessing is finishing there. Then next step is vegetation segmentation, which logically also connected to the preprocessing, but in fact, it's already working on the prepared data. And consequently, we are familiarizing ourselves with the configuration management, so how to operate with the config file, which is automatically generated during all the steps of processing. And then we are learning how to use this configuration into different hyperspectral images preprocessing pipelines. So then we are applying this configuration also for the batch processing, and the final step will be demonstration of potential steps which can be done afterwards, with also some teaser hinting on how it can be improved. So if we are moving to the introduction, introductory part is explaining what the hyperspectral library from PSI is useful, so what it can be used for, what main functions it has. So we have here detailed description of the structure of the repository. So we have 3 modules. Score module, which is operating with the, directly with the hyperspectral images. So it's so-called HS image and MS image objects, which store the hypercube plus metadata plus some basic methods necessary for working with the hyperspectral images. Then second module is preprocessing module, which is storing all the methods necessary for the preprocessing pipeline. It uses all the logic necessary for the chaining of this method to the configuration management and for the some functions for visualization. So the last one is utils module, which has most of the visualization methods. It uses so implements some tests. It checking inputs outputs. It has functions for checking input outputs operations, and it also implements some supplementary tools which were not suitable in other modules, which are maybe not 100% obligatory for the analysis, but useful for visualization or some additional operations. So then there is short introduction into the file format we are using in hyperspectral images, mostly. Software, so hyperspectral software from PSI, our cameras are automatically generating, taking images in this format. It's a called environment for visualizing images, NVI format, which consists of 2 parts. So first part is so-called header or HDR file. It has all the metadata describing the resolution, dimensions of the hypercube, and describing all the indexes of the wavelength. So we definitely know which wavelength we have at which index, at which order. And the second part is essentially the hypercube itself, which is stored in BIL files, so called band interleaved by line. So it's just a piece of data without any metadata. So as I said, metadata is applied in the header file, and each header file is connected with the respective BIL file with the same name. So only the end of the file with the file format is different. So like two files are comprising one object. So then we are moving to the workspace preparation. First thing first, we need to install all the libraries required for the smooth working of the pipeline. So first row is installation of our HCI PCI library. In the next month, I hope, I will publish it to pip, so it will be easier to install. Currently, it should be installed from the GitHub. But essentially, this installation is not so much different like the second one. So, and additionally, if you want to check out for the repository itself, you can just copy this address and go here, look how, and you are accessing the repository. Concerning the repository, it is you're free to suggest modifications, you're free to suggest improvements. So if you're interested, if you're curious, please go through, check how the library is working, and don't hesitate to propose your improvements if you see any errors or you want just to implement some new methods. So here we got our green check mark. Okay, hello. So here we get our green check mark, which is saying that the cell execution is ready and it was successful. So we can move to the next step, and the next step we are importing installed libraries into our environment. So here is a short description which library is doing what. So we have here, shortly speaking, the libraries for the visualization. We have here our HCI PSI library. We have here some settings for the visualization, some settings for the hiding the warnings which some packages can show to reduce the size of the output. So we have here some system libraries like OS for manipulation of the files, for reading JSON files, for copying files, for the usage of principal component analysis, or the SNA dimension reduction or embedding. So yeah, you can just go step by step and Googling which library is doing what, and most of those are just standard Python libraries. So next step would be mounting of our Google Drive. You see here the comment that it's only for Colab users, so it's our case. If you are working with the local installation, you don't need this point, and it will probably cause the error if you execute this cell. So here we are allowing Google Colab to connect to our Google Drive, considering that they're connected to the same account. That should be considered safe, and you see all the code. So, and then we are storing our path to data into variables. So instead of writing these huge strings of path strings which represent the path to our data, we can just store them to the convenient variables and continue with those. So this is the path to the main file. So main folder where everything is stored. In my case, it's called EPPS dummy. So in your case, in the file which is shared with you, this dummy will be deleted. So I just don't want to modify the originally shared workshop, so I'm doing it in the separate folder. In your case, this will be just EPPS 2025. So, and then we have the path of data folder where we have our data for the measurement. Then we have path for data for the references. Then we have path for the configuration folder, and then for the folder where outputs will be stored. After everything is saved, we are visualizing content of data folder just to show that we indeed read correct directory. So then we are out of the files saved in the data folder. We are selecting one on which we will be testing our preprocessing pipeline. So this image will be, so the path of this image will be stored in this variable. Then we are also saving the path for the white reference and dark reference. What do we need white reference for? I was a little bit mentioning in the main body of the workshop. Yeah, so let's execute this. But we'll turn to this just a minute. So, and here, kind of important step, we are initializing the preprocessor object. So preprocessor object is the heart of the preprocessing pipeline. It's the object which stores hyperspectral images inside itself. It's the object which traces all the steps which were taken during the preprocessing pipeline. It stores also visualization methods and numerous metadata attributes of the hyperspectral images, and essentially addressing different methods and attributes within this preprocessor method. We are going to go through the whole workshop. So essentially, inside of this object of the preprocessor object, we have 90% of all the methods we will need for this workshop. So, so to create new preprocessor, which will which will represent the preprocessing itself, so we need to initialize it. So new object, for now absolutely empty, with empty configuration file with no hyperspectral images loaded, is created in the cell, and then on the next step we can load the image. So this is the path we set for our image of interest here, and with the command load image we can load it. Probably one more thing which probably important to mention here is that when you initialize the hyperspectral preprocessor, you can set the output format. So whether you want to be informed about every step of preprocessing which is taking place or not. So for this, you have this parameter verbose. So if you want it to be very communicative and provide you a lot of information, a lot of output with concerning every step which is done under the hood, you can set it to true. Otherwise, if you don't want to get excessive cell output and yeah, so and if you want to have your environment a little bit cleaner, you can set it to false. You can play with this. You can set it to false. We can set it to true settings for the second preprocessor, which will be defined just soon, and you will see the difference. So now let's return back to the loading of the image. So we will load our image. So the method which has only one parameter, the image path. You see that it returned the preprocessing object, which in this case means that everything went smoothly. So we have new object. Good. And then probably some information about the hyperspectral image would be useful. What we loaded. Hyperspectral image is another entity, it's another object within the preprocessor, which has essentially the structure of hyperspectral image which you've seen before. So it has the metadata part, which is stored in the hyperspectral image meta parameter, and it has hypercube part, which is stored in the hyperspectral HS image parameter. So you can explicitly call for the hyperspectral image index, which will return you the list of all the wavelengths. It's very useful for example, plotting the image, or just plotting some particular vector, particular spectrums. Then you can also retrieve the name of the original image and essentially have much more of those parameters, which you can visualize or check after you import the image itself. So yeah. So here we, in this step, we will create new hyperspectral image. We loaded it in the preprocessor part, and now we can retrieve it from the preprocessor object by calling preprocessor image. So, and our hyperspectral image is stored here in this variable. By this command, we will print all the metadata. Here, we'll print all the wavelength. So we'll list the wavelength, and here we will print the name, original name of the hyperspectral image. And you can see that everything was executed correctly. So here we have the dictionary with all the metadata, including wavelength as well. So here you have the list of wavelength indexes. And as the last option, you see that we are getting the name of the original image. So information that we got our object right. Next, two functions are quite similar by the syntax and output, but they serve completely different purposes. So second function, first function is get RGB sample function, which generates pseudo RGB representation of the hyperspectral image and returns this representation. So by executing this command, you generating RGB representation of the image and save it and output it like a three-dimensional NumPy channel. So because as I mentioned before, it perhaps not so easy to visualize hypercube sometimes, and it can be useful to visualize it like a video. But if you're doing some plotting, then perhaps it will be more useful for you to have it like just two-dimensional, colorful image, so you can see which changes were conducted during the process. It will be not conducting all the information which is stored in the hyperspectral images, but sometimes it's more than enough. And that's exactly what we need from the. That's exactly what we will be doing with the get RGB sample image. So this method is requiring several arguments. First two, normalized and correct, are normally used for the improvement of the quality of the image. And that's exactly the reason why we want to be using them. We want to see how our raw image is gradually changing during the processing. So we're setting both to false. Then we're setting argument show to true, because this argument is responsible for the showing or hiding the visualization of this false RGB image. Then we are setting the title for the image. We are setting visualization of axis to true. And here there is one more important parameter is repeating. As I mentioned during the introductory part of the workshop, we're using, in the field systems, we're using track-through, which has limited minimum speed, and so we cannot go below some limit of the image acquisition speed. And when the light conditions are good, so we have sunny weather, this speed of scan is sufficient and we're getting nice image resolution. But in this particular experiment, illumination, so light conditions outside were quite cloudy, and the intensity of the light was not sufficient, and therefore we, with the exposure time set to minimum, the speed of track-through was too fast, and our images got a little bit squished in y direction. That's why for the visualization, and only for the visualization, so we are not modifying the original hypercube, we need to add this repeat parameter, which is stretching, essentially the stretching the image several fold for the better visualization. So if we set this parameter equal to 10, we'll get it 10 times stretch in y direction. And if we don't need this parameter, if we're working with some lab generated images or the field images from the good environment, we can not mention, so not put any argument to this parameter at all. So we execute our visualization. This RGB image is then stored into the raw RGB variable. And we can continue on. We see that the image looks weird, so the white balance is completely off. We see that we hardly can distinguish between the background and the green vegetation. We don't see Teflon reference almost completely, so some dark band here. And Teflon reference should be supposed to be the best illuminated part of the image, so it should be white and shiny. So here we have quite challenging light conditions. And we see that we have heavy shadowing of several flowers. So, and with all of those, we will be fighting in the next steps. But before we will start real preprocessing, I would like to show you also one more useful method. It's a spectrum probe, which is also visualizing the false RGB representation of the plant over the image, but it returns not the raw RGB image, but the spectrum. And if you supply it with the region of interest of particular spectrums with their names, the format of dictionary, it will return to you also the dictionary, but not only with name, coordinates, but also with the spectrum of this particular region of interest. So this spectrum probe gets almost the same arguments like the previous get RGB sample function. So same function, same argument for repeating, same function for title, axis, normalizing, correcting, but also gets rows. And rows are supplied as the dictionary, where we have every row supplied with a name. Then we have coordinates supplied to the slice objects. So the first coordinate, the first object here is the row, so y direction, y-axis. So this is coordinate of starting, this is coordinate of ending. Pixel, then we have x-axis or width. Again, just column if you wish. The column where it starts, so you can more or less have an impression which ones, how to set those rows by those labels of the axis on the get RGB sample. So you can map your rows looking at this image. And then you also can slice this spectrum probe through the spectral axis, though we don't need this. We want to see all the spectrum, so we just don't supply this information on the third axis. And then with comma, you can supply as many rows as you want, as long as they are named differently. So we are visualizing our, so we are saving our rows in the format which I mentioned below into the variable to make our syntax a little bit more intuitive and concise. And then we are visualizing the region of interest. So I've selected the illuminated flower. I selected the shadowed flower, I selected the region of interest for the background, and I selected the side Teflon region for the reference. You see that illuminated flower here by far is the most, has the strongest signal, which means that everything else except of him is probably currently in the shadow. And let's see how the evolution, how those spectrums will be changed after every processing step. Yeah, so here you see the important note. And this is very important to mention that our preprocessor object doesn't have undo method and doesn't store the previous state of the hyperspectral image. So if you want to play with the preprocessor steps, you need to create a copy of the preprocessor object. And then after the copy is created, you can use this copy to experiment further with different processing steps. For this, I suggest to use deep copy. So we have both objects, the original and the copy, completely separated, so we don't modify the original one. And then, yeah, you can move on for the experimenting. And the first step of the sensor calibration is a sensor calibration, which is used for the compensating for the sensor and even sensor defects and sensor artifacts. Every hyperspectral sensor has the regions where the signal is lower. So you can see it on this part. I'm visualizing the supposedly flat spectrum of the white Teflon. So you see that our spectrum has regions where its sensitivity is lower, is on the side, where sensitivity is higher in the middle. You see different dents and dips and peaks, which are getting from the pixels of different intensity and other optical aberrations. And we need to compensate with this. For this, before you start your experiment round, or essentially consider for every imaging, you need to take one image of white reference. So white reference in our case is a Teflon plate, spectrum of which we know. And then you are compensating, so you're applying the sensor calibration function, which is transforming your spectrum to compensate for this, for the difference in the sensor sensitivity. So essentially, we are launching our sensor calibration. We're supplying this method with the white reference path, dark reference path, and we also can ask it to clip to the particular range, meaning that we will get rid of the outliers bigger than the 10 sigma from the median signal. So big spikes, or really big spikes, should be removed by this. And then we can, sensor calibration is conducted here, and then we can visualize the result. Yeah. So here you can see that results changed, so that the RGB representation of our image changed quite significantly. And now we at least can say that the color balance is getting to normal. Spectral shape is also getting to normal. That's exactly how the spectrum of calendula flowers looks when you measure it with the spectroradiometer, like the polypan or something like this. So you already can recognize this shape as correct one, and well, this is a good sign. And now we can also compare what happens to our region of interest. We can see that the scaling was not included into the sensor calibration, so it modified the shape of the spectrum, but not its scale. This will be done in the next steps. And we can move on to the next step. And next step will be the spiking. And you probably can notice why this step is important. Because, as I mentioned before, the spectral information in veneer and sphere range is usually encoded in the smooth low frequency curves. So information about any pigment, information about the drought stress, whatever kind of stress, is usually at reflectance of the water bands, different nitrogen dependent bands, essentially all the information we have here, meaningful information. is encoded into the low frequency curves, meaning that everything you see like spiky and sharp here can be considered rather noise. Especially you can see that the second sign of this is that we see for every ROI these spikes to be located in the same location, to be more or less the same height compared to its surroundings, which strongly suggests that this is really noise which we should get rid of. And the first step would be despiking, because if you apply the traditional smoothing right now, this spike can affect it quite a lot. And actually, it's probably even not the best idea to apply smoothing right now, because we will be doing also some solar correction. And it's better to smoothen our spectrum after the solar correction is done. So now, just not to mess up with the solar correction, because solar correction can be also distorted by those high spikes. We just getting rid of the highest peaks and dips using the spike detection method, so remove spectral spikes, which takes 3 arguments, is the context window, so how many pixels around every particular wavelength it analyzes, then key parameter, which supplies the parameter for the number of standard deviations is used to set a threshold. So simply speaking, the function is analyzing the surrounding of each particular wavelength. And if it finds any value which is getting higher than the threshold of 3 sigmas, it replaces this with mean or medium. And this final argument replace is responsible exactly for this. So let's execute our cell. Yeah, you see the notice that it can be time consuming because we are doing double iteration here. Essentially, this is the process which is quite similar to smoothing, so you cannot vectorize it completely. So we're going through every pixel of every pixel of the image, and then through every wavelength of this spectrum, and applying quite intensive for computation the spike removal method, and we'll continue as soon as it's stopped. Okay, here it is. We can visualize what happens with our spectrum, and you can see that visualization didn't change much, and our spikes are mostly gone. So you see some small ones, but probably it's because we set the sigma or the window low enough to allow the small spikes to persist. You can play clearly with the both parameters to find the spiking which will be suitable for you. Okay, and let us move on. The next step would be solar correction. Necessity of solar correction was already mentioned in the intro, but let's get back to this again. Solar correction is necessary because during the imaging, the solar spectrum is changing quite significantly, especially if the imaging is taking a long time. So you can see that at the noon, the spectrum of sun, the blue line, is quite different from the spectrum of the sun at the 6 a.m. So, and those changes are quite significant, and they are not only the change of the scale, change of the albedo, it's also change of the shape of the spectrum, and we need to compensate for this. And for this, we use the second reference Teflon, which is mounted right under the hyperspectral camera and present in every line of the push broom scan. So essentially when so here's the camera objective, here this white side Teflon reference, and then we can normalize our image, every row of the image to this Teflon reference. So essentially we are getting not only per image solar correction, but per row image correction, which is very convenient because even during one scan, illumination conditions can change. So, and for the solar correction, we need also some reference spectrum, some kind of ideal Teflon spectrum, which which is used like consequent reference. And for this, we have two options. Option number one can be that we simply take the reference spectrum from the first image we're analyzing right now and take it as a reference, and then all others spectra, all other images which will be analyzed will be recalculated to this. But considering that, and it would be probably the most rational way to start because it's fast, efficient, intuitive, and clear. But here I want to show you actually what this solver transformation is doing with the spectrum. And for this, we will use a little bit different tool. We will use create reference to Photon library function. What this function is doing, the function is extracting the site Teflon image spectrum from every image in your hyperspectral images folder. So all the images which you stored in your data folder will be analyzed, will be loaded, they will be processed with the preprocessor to the stage of analogous to your current preprocessor stage, and then Cyteflon will be extracted, a library of the Cyteflon spectrum will be generated and then averaged, and this average value will be saved in this variable in the output. So this create reference Teflon library method takes path for the where it can find the hyperspectral images or your data folder. Then it needs the white reference path because it's also doing the sensor calibration. It requires also the coordinates of the site Teflon. That's how you recognize the methods which are necessary for the field usage only. For the lab conditions, you don't need to do anything with this. So solar correction should not be applied for the lab usage, and therefore on the lab images you usually don't have this site Teflon, and yeah, this part isn't for you. So for the field images, it's better to supply coordinates of your reference. So if you use different kind of reference, if you use different kind of reference, you can change these coordinates if you will. Yeah. So, and here's an important note. The method is going through a lot of folders, so a lot of images in the folder, and if the amount of your images is big, it can take quite a lot of time. Therefore, we won't be launching it now, not to occupy half of the next workshop of just processing of this Teflon library data. So those two cells are dedicated for the extracting of the reference Teflon spectrum and saving it locally. So we will jump over and we'll use the reference Teflon spectrum, which was already pre-extracted. So before the workshop, I launched this method and extracted for you, so you can find it in the same shared Google Drive or OneCloud folder on this address. So to 2020 APPS output reference to Photon spectrum. Yeah, JSON. And then we can just open it, get the message that reference to Photon spectrum is loaded, and then use it. Actually, yes, here is quite important thing. Here we have the supplement function from the utils model for the plotting spectra altogether. If you remember the visualization method we were using before the spectrum probe is generating the library, that is generating the dictionary with the spectrum. Yeah, you can actually see here quite nicely how it looks. So it's output of the spectrum probe is the dictionary where we have the row name, then we have the spectrum recorded, and then we have the slices, and then we have the indexes. So essentially, we are getting quite comprehensive information about these rows. And the tool for the spectral visualization, this plot spectrum, is using this format, those dictionary for the visualization of the spectrum. So you simply can supply output of the spectrum probe method directly to the plot spectra, or you can, as it's done here, just drop everything what you don't need and leave in this SpectraProbe derived dictionary only those pieces which you need, and then you can visualize those. So what we're visualizing here. Plot spectra is taking the list of dictionaries with the spectrum which you want to visualize. In our case, the one dictionary was from the current cytoplan, and the second one it was from the reference cytoplan, which was exported exactly in the format to fit into the function. So then we have, here we mark the spectrum names, reference, and the current image, and supplying it with title and scaling. We can, so here we are scaling to see the shape changes between those spectra, but we can set this variable to false and to see the real difference between those two spectra, which will be, yeah, you see that we have the current image Teflon much less illuminated than average side Teflon. And our solar correction will compensate for both of those changes. So it will change the, it will scale the image to the reference Teflon and also will modify the shape and then apply this information to every pixel of our current image. So when the reference side Teflon spectrum is found, we can supply it to the solar correction function, which takes again the coordinates of our side Teflon and the current image, the reference Teflon spectrum, which is stored in the dictionary format, as it should be here, and the smooth window. So what's going on behind? So why smooth window? What smooth window has to do with this? As I mentioned before, the valuable text information in the linear spectrum is stored in the smooth low frequency curves, and we here we also don't want to implement any noise which we can find in the side Teflon of the reference spectrum. So we want to implement only transformation of the trend. So we want to apply transformation of the shape of the spectrum itself. So we are applying quite heavy smoothing to the both Teflon and only so reference and current Teflon, and only then we apply we calculate the transformation formula. So then yeah, so here solar correction is done. You see here the coordinate of the side Teflon or the current side Teflon. So here we have the reference spectrum array extracted from the dictionary, and then we have the smoothing window. And let's see what is information done to our data. So visually it looks almost exactly the same as it was before. So maybe I just see some warmer tint to the image, but this is a RGB representation, so it's quite poor representation of a ridge hyperspectral cube. So maybe it's just not how it looks. Let's see forward. And the spectrum shape looks more or less the same. So the solar correction was not so big, maybe. Let's check it out. Let's plot our spectra nearby, and for the plotting the spectra, I selected the illuminated flower ROI from the solar corrected image and the same ROI from the uncorrected image. So I explicitly extracted them as arrays from the respective dictionaries, which we got from the, so this is output of the spectrum probe function. So just to make it clear. And the second one is the same output of the preprocessing spectral probe, but just for non-corrected region of interest. And if we plot those illuminated flower spectrum near one by one, we can see that actually the changes were quite substantial, I would say. So we indeed get, just go a little bit higher in the red range of the spectrum. So that's probably means that this red tint, which I've seen here, As I mentioned here, it's really something which takes place, and then you see that we continue with this change of the shape of the spectra also into the far red range, and also just modify a little bit spectrum at the end. This is not so much relevant, and actually this can be a little bit an artifact. We'll address it in the next steps. And now we are moving for the spectral smoothing when images are finally sort corrected. And as I mentioned, the spectral smoothing is ultimately serving the same purpose as the spiking, maybe just a little bit more radical. So we're applying it only here because I just wanted to store it as safe as much information as possible before any physical transformation of the spectrum, like calibration or the correction will be applied. So we have the real spectrum. And now we finally can get rid of all low frequency noise, which still remains in the image, like those small peaks and data noise. So we are using the Gaussian smoothing for the spectral smoothing, which, according to my experience, is one of the best ones for this role, and which is probably one of the most intuitive ones. So with Savitsky Gallet, you need to set two parameters here with the spectral smoothing. Essentially, there's only one important parameter, which is sigma. And if you imagine, so where this sigma is going from, if you imagine the Gaussian distribution, you know that the distance from the median is defined by is very often like described by the number of standard deviations. And at sigma 3, if I'm not wrong, we have something like 99.5% of data within the distribution and only of marginal of 0.5% or something like this outside of the distribution. So essentially by setting the sigma, you are setting the width of the spectral smoothing window. The bigger it is, the more smoothened will be your spectrum. Just the one parameter, convenient and intuitive. So the smoothing part should be recorded once again. And here we will cut smoothing. So for smoothing, we are using Gaussian smoothing because, according to my experience, it's one of the most intuitive and best smoothing methods for the spectral data as well. It has essentially only one important parameters, which is quite easy to interpret. It's sigma parameter, which essentially describes how big window you are analyzing for the smoothing and how strong your smoothing will be. So the second parameter reflect is essentially telling you only how the marginal part of your spectra will be processed, because it's quite known problem that when you smoothen your spectrum, your edges are getting uneven smoothing compared to the middle regions, and with this reflect function you can address this as well. So let's do the Gaussian smoothing. It's quite a quick process, and then visualizing our image. As you can see, the RGB representation of the image didn't change much, but we've got nicer, smoother curves. So then the next part is exactly what I mentioned, that will be where we address some artifacts at the edges, is spectral range cropping. So as you remember in the previous step, we've got some spectral shape difference at the edge, and additionally to this, it's quite widely known that spectral sensors, so the hyperspectral sensors, they have lower sensitivity at their margins, at the edge of the spectra they're sensing, and therefore it's quite common practice to get rid of 15 marginal wavelength from other side. In our case, it will be from 350 to 400 or 410, so the range of 40, 60 nanometers, and the same cut from the near range. So the preprocessor, the cropping is done by crop spectral range function, which takes the starting wavelength, finish ending wavelength, and cuts everything what's beyond this range. Instead of wavelength, you can use the band order parameter, but probably more intuitive it will be just using the wavelength. So we're conducting the spectral cropping and visualizing our spectrum afterwards, and we see that our spectrum indeed is now shorter than it used to be. So the last step of normalization of preprocessing proper, so where our images will be transformed, is the normalization spectrum. And the need for normalization arises from the observation that here we still have illuminated flower quite much different from the shadowed flower. And we want to build, so our ultimate goal for the study, which I was showing in the introduction, and for this workshop, is to build a classifier, which means that we should extract as much as possible of pixels from every genotype, which will be as homogeneous as possible. So we need to shrink this difference between illuminated and shadowed plants. As I was asked during the workshop, and which was very good comment, that not always this normalization is needed, because sometimes we need to build model for the prediction, for example, of the chemical content. So we need to build a regression. And there, in the regression, those difference in the albedo and the intensity of the signal, in the scaling of the curve, they are actually can be informative by themselves. So in this case, I would suggest just creating some model which will be detecting the shadowing and getting rid of the shadowed pixels altogether, and then within the normalized pixels, just within the illuminated pixels, just don't normalize. So taking all normalized pixels like they are and continue without any kind of scaling. In case that we are building the classifier, the step is necessary because, as I said, we need as many as possible and as homogeneous as possible data. So we will try to compensate for the shadowing. And for this, we have several options. Our normalized spectra function takes 3 arguments. The first argument is method. The method can be selected to normalization. So these are normalization to particular wavelength. So the whole spectrum is essentially divided by the particular wavelength, which we can define in the second one. So 2VL, where we can set our wavelength number. Or if different normalization or standardization method is used, it can be either just set to L2 normalization or to standard normal variate standardization, or to the robust normal variate standardization. So for the matter of efficiency, I would say that SNV, so standard normal variate, or RNV are the most efficient ones because they also center the data and they standardize them to the unit variants, which help us significantly to the dimension reduction, which increases the efficiency of consequent principal component analysis if you want to use it. And different embedding methods, for example, if you want to just work with the CNNs afterwards, it also will just improve your result a little bit, especially with shallow CNNs. But if you want to visualize your data afterwards, so visualize the image, which we want to do, it's better to work with the normalization, so L2 or to normalization to the particular wavelength, because standardization, as I said, it centers the data around 0, so we get negative values, and visualization of negative values with image with negative values is not so easy. So we are using for this meta the normalization to a particular wavelength. You can play with your results with SNV or L2 or RNV as you want. So, and let's visualize what normalization did to our image. And here you can see that we actually did quite some progress concerning the homogeneity of our images. So even so, normalization is always a very pleasantly looking step where your image start to look very homogeneous. So you see that difference between the illuminated and the shadowed flowers became much smaller, though this image is still present. So you see that while they're scaled to be of the same size, the shape is quite different, which is totally okay. First of all, because the calendula genotypes have quite a lot of inherent variability. So even from one plant, you can get quite some different flowers, and their spectrum will be explainably, quite logically the same. Not the same, but quite logically quite different. And the second point here is that the shadowing, it's not solely the scaling effect. So you have the reflectance from the green vegetation, you can get reflectance from the walls of the tractor itself, of the camera, from the soil. So you have a lot of different effects which can be partially attributed when you're using methods like SNV because they are dealing with some degree of light scattering. But essentially, to get rid of it completely, you need some much more complicated model, which we won't be covering here. But we've done what we could, and I would say that is already quite good what we could extract. And let's continue on with the steps which are following the preprocessing. After our images are ready, we can move on for the segmentation of plant pixels, and we are going to select probably the easiest way as segmentation of vegetation index. Yeah, in our introductory part, I mentioned that we were using the machine learning model for this part. So yes, we were segmenting the vegetation indexes, but we were using then the machine learning model to automatically set the threshold for each vegetation index automatically. Here we will be using the manual thresholding, simply because setting of the interactive annotation would take us probably up to an hour or even maybe more time on top, and we don't possess such resource. So if you are interested in this part, so with more interactive, with more interaction with your original data, we can organize an additional continuation of this workshop where I'll show you how you can annotate your image manually and then supply those annotations to the segmentation model, so you don't need to play with your thresholds manually. So what is what's an idea behind the threshold, so behind the segmentation here? The first step, the function extract mask generates 3 vegetation indexes maps, which are one-dimensional 2D images, and then it takes 3 arguments from the user or from the configuration, as a matter of fact, and applies those thresholds for these maps. As a result of this thresholding is a binary mask for every vegetation index, which then they combined, and out of the combination of those 3, we're getting the ready plant mask. The vegetation indexes here are selected by the empirical testing, so which ones would be better for our matter. So PRI here is used for the segmentation of flower pixels. It's quite good with this, but we are getting quite some artifacts. And to get rid out of these artifacts, we are using NDVI index to subtract all the green vegetation pixels, and blue NDVI index for subtracting all the soil pixels. Surprisingly for us, this index was quite good for detecting the bare soil pixels. So then, after detecting the flowers and subtracting all the artifacts, we can also apply the minimum pixel size spectrum to get rid of some single or like small artifacts, which sometimes can arise on our segmentation. And yeah, we also can visualize the result. And this visualization can be repeated for the reasons which I described above, that sometimes we need to stretch our images. But again, here it doesn't affect the original hypercube, just visualization. So here we have some preset threshold, which were empirically found before the workshop. Yeah, please pay attention to this version of the workshop and the library. There is still this vegetation index for the soil, pixel soil detection is still called HBSI. That's not correct. Initially, it was idea to put here HBSI, but then I figured out that we have a mistake, and real HBSI should have the severe range. One of the wavelengths should be in the severe range, which we don't have at our disposal here. So index named HBSI here is, in fact, is blue NDVI. So in the I'll fix it in the repository, and then the name of this parameter here will be changed to blue NDVI, BNDVI threshold. But for now, just knowing that essentially this is blue NDVI, we can continue on. So we supplied thresholds for our segmentation. Here is already not a real preprocessing, it's already segmentation, so we don't modify our hyperspectral images themselves, so we can play without being afraid of modifying anything in the original data. So we can play with different vegetation indexes parameters with different thresholds. So we can see how different thresholds will look. So you can see that if you are going into lower values of PRI, you're getting much less pixel segmented. So if you are selecting a much wider frame for the NDVI, you're getting essentially all the greenness inside of your mask. And if you are getting with the blue NDVI with a wider frame, you're getting essentially all the swell included into the mask. So you can play with this as long as you want. We will return to the pre-selected thresholds. So we will execute this step again to write our selected threshold into the configuration. And then we are moving to the next session, which is a saving configuration of the preprocessing pipeline file. So here you should consider that the segmentation itself is already not real preprocessing. It's already some processing step which is applied on the ready images. But here, for the logic's sake, for the convenience, it is included into the preprocessor. So when you're doing the segmentation, it's also recorded into the preprocessor object, and it's stored in the configuration. And when we call for the configuration file, we can see all the steps and the order, how they were applied, stored in the configuration parameter. So you can see that here the first was the sensor calibration with all the parameters, with the path to the white reference. Then I have spike removal, throw correction, all the steps. And you can use this configuration for the application to the next image. And just to emulate real live data, how it usually works. So you usually do this calibration and application to different images in different files. So let's save our configuration to the local file, to the local JSON file with the save config command. Yeah, so here it's stored locally, and then we can reuse it with the different image. So and now we can use this stored configuration file for the applying the old preprocessing pipeline for the new image. So for this, we need to visualize new, visualize new preprocessor object. So it will be preprocessor 2. Yeah, we're switching. Okay, let's do it actually verbose just to see how it looks when the preprocessor is talkative. So we're initializing the new object. We are loading new image from different genotype into this object. And if, you see that output is already bigger than it used to be when verbose was false. So now the preprocessor will be sending us much more technical information of which steps we're taking, what is the current status of the image. It's informative, but sometimes when you do it repeatedly several times and you're absolutely sure that your pipeline is working and you just want to apply it, it can be quite messy. So now we can control the status of the configuration, and configuration is empty because we didn't do any steps for our just loaded raw image, and we didn't import any configuration from outside. We select, I've pre-selected the rows of interest on this new image. We store their position into the rows variable, and let's visualize how our new image looks. So you see that here it's definitely different genotype. You can see that here we have totally different color. And what's interestingly, you can see that the illumination conditions in the range of interest are drastically different from the first image with the yellow flowers, which we were analyzing. First of all, the difference between the flower, illuminated flower, and the shadowed flower is not so big as it used to be in the blue one. Second one, the cytophlone intensity is finally, just as it should be, is illuminated to some extent, which is like reasonable. And it means that this size of the cytophlone here is illuminated. In the previous image, it was obviously shaded. One more region for the solar correction we applied, and you can see that background, so the soil itself is also illuminated much better. So this image will have substantially different solar condition to which we will be compensating. Nice. So let's continue, and here we will load the configuration file from the which was stored locally from the previous preprocessing pipeline, and here we'll just visualize what's inside. And yes, so loading was successful, and configuration of the new preprocessor is now identical to the old one. And then with one simple command, we can run the whole pipeline for the second image. The parameter which this run whole pipeline method is taking is configuration override, which is optional and which can be useful if you want to apply the loaded pipeline which you took from the previous preprocessor method, but modify something a little bit. For example, you can supply this argument config override with the dictionary of the type normalization method, and just supply here the parameters for a different method, and this will be enough. So you can supply it with the dictionary of this format, and this run full pipeline function will apply all the changes from the loaded configuration, except of the normalization, where it will use the dictionary, so the parameters which you send with the config override argument. So then we have the extract mask flag, which is automatically set to true, which is automatically doing the vegetation pixels segmentation. So here we will run the full pipeline, which will take some time. And after everything is extracted, we'll return back. Yeah, actually here you can quite nicely see why this mode is initialized by verbose and what it means. You see that we have here detailed description of every step which was done. And the report where it took how it loaded the image, what operations were done, and how they were conducted. So this step can take up to 10 minutes, and after it's executed, we'll get back. Okay, so that was a little misunderstanding. The processing of one image, of course, doesn't take 10 minutes, so it took just a minute. So we are back, and here we can just actually check how every step of processing was done, and we can see the final result of the segmentation. So we see that the mask is actually quite accurate, and we can see the masks and the maps for all the vegetation indexes. So nice, we see the segmentation, but under the segmentation, we don't really nicely see the plant itself. So let's visualize our map and spectra. Yeah, and you can see that the image looks good. The cytoplasm is shining, the background is background, actually more green vegetation than background, but anyway. And so here we have flower and shadowed plants. So here we can check the spectra. And as we've seen before, the difference between the illuminated and shadowed flower spectrum is much lower than in the previous image. And here it also can be seen that the shape of the spectra is not so different between those, because even initially the difference between the shadowed and illuminated was not so big. So we can explicitly force our pipeline to extract the mask, as if here in the previous step the extract mask flag was set to false. So we don't need to do it, we just done it. And we are moving to the batch processing. So batch processing is done with the process folder function, which takes four main arguments. The first one is the folder path, which will be processed. Second one is the configuration path, which will be loaded. Then again, verbose parameter, and then parameter if we want to return directly to data frame. This parameter is quite useful if you don't want to store all processed images in the RAM, because datasets can be quite big. And with this parameter, the method will be analyzing each image one by one and then rewriting them. So you won't get stored hyperspectral images in your memory, and you getting only one data frame output, and essentially your RAM won't be you won't have problems with the too many resources occupied. But also pay attention that this parameter actually changes the output type which function give you, because if this parameter is true, the function will return you a data frame. If this parameter is false, the function will return you the list of processed images. So we will set it to false to then extract the data frame separately, just to demonstrate how it's done. And let's execute our pipeline to every image. You see that we're getting, again, verbose report on the every step conducted, and then I immediately launch the function for the extracting mask spectrum to data frame, which will use the list of processed images, which we'll get as output here, and will extract the masked pixels into the nice and arranged data frame. So when both of those functions are executed, we'll get the message that execution is over, and we can continue on. The execution can take up to 15-20 minutes together. Yeah, you see that we're getting quite an extensive report of what was segmented with which parameters. And yeah, maybe just a few words about the method extract mask spectra to df. We get so it asks you for the list of processed images. It asks you for the path where you want to save your data frame. So this is optional one, but quite convenient. So you immediately get one object in the memory of the data frame, and you can then save it. And so here the parameter which asks you if you want to include segmentation as the final step. So if you want to store also the masks. And yeah, so that's essentially it. And we're waiting for the function execution. And when the execution is over, we will continue to the visualization and the next steps. And now, when the processing is over, you can see that altogether it took how long exactly? 12 minutes, not so bad. We have report and we have our extracted spectra ready, nicely packed into the data frame. So then we automatically stored the data frame at this location, and we can visualize it. So we are here grouping our resultant data frame by the label, which means by the genotype name, subtracting the mean value, rotating it, and then plotting with the given figure size, marking it respectively, and just visualizing true, visualizing grid and legend. So, and you can see that clean spectra look quite good. With this extract, with the function extract max spectra to df, we can actually also extract the background pixels, which is a category for all pixels which are not segmented by any other mask. And this can be quite useful, for example, in cases when we want to train some model for the segmentation, for the vegetation segmentation task. There you will get automatically the spectrum of every class, of every genotype, together with the spectrum of the background. So you have enough of data to train your model on. And then we are moving to the principal component analysis part. This part will be extremely short, so I just want to visualize what potentially can be done with ready data. And yeah, so we are loading our saved data frame. So essentially the logic here is that every section can be launched independently from the previous one, so all the data are saved locally, and you are not obliged to execute every cell before, except of imports and libraries installation, to continue working with this data frame. So what's happening here when we transform our data to PCA? PCA is beautiful methods which is finding the axis within the data with the highest variability, with the highest variance, and projecting our data points into those axes. So essentially here, as we were speaking in the introduction, we are projecting the data points from 2 dimensions to 1 dimension, and the one with the biggest variance is marked here by this violet dashes. So in our case, we are projecting our hyperspectral data, which of those have more than 400 nanometers, 400 values, to just 10 principal components, so the 10 axes with highest variance. And before we go for it, we are dropping the background category from the pixels, because while it can be useful for this consequent segmentation training, we won't be using it for segmentation today, and we'll be using for the classification solely, and therefore they may quite substantially affect how the data distribution will look after the PCA, because the background difference, the background spectral is different quite a lot from all the planned pixels categories, and there is quite a big chance that the first first several components will be dedicated solely to the difference between the background pixels and the flower pixels. Considering that our aim is to train the classifier, means that we want to extract most informative threads for the distinguishing between different classes. This background category can introduce quite a lot of distortion in our data. So then, after dropping the background, we are separating our data to the informative part, to the data part. This is labeled like X, and the label part, which is marked like Y. So then we are initializing PCA objects from a CKit library. So it's already not part of the preprocessing pipeline. And then we are fitting and transforming our PCA with our data. And then we can visualize, so we are printing the variance ratios for every of those 10 principal components. So let us see. And yes, so everything is done. On the last two steps, we're creating new data frame with those 10 principal components as values and adding the label column from the original data. Then when we finally have our data transformed, we can visualize them. And the most common way to visualize the PCA data is, of course, plotting them, scatter plot. So for this, we use the methods from the utils module of HCI PSI library. So visclust 2D, which serves for the visualization of 2D clusters, and it accepts essentially the data frame together with labels, and then the list which marks which principal components we want to visualize. So if we visualize the first two principal components, and as usual in Python, the counting starts from 0, so 0 means first and 1 means second, we see that we have got the data. Actually, we've got a picture very similar to what we've got when we plotted the spectra. We have two spectra which are very similar to each other. So L15 and L25 are close to each other, and L26 and L4 are so similar that you barely can distinguish them. And this same information is conveyed then into the first principal components, where you can see that L15 and L25 are similar but separatable, especially if you play with different principal components. Now we're visualizing only two. And that L4 and L26 are really hardly distinguishable. So if we go for different principal components, potentially this image can get a little bit better. Yeah, but it's really the matter of the other processing. So you see that we are getting some outliers here, so probably we've got some background pixels included, and they create in this wide range. So essentially, this is how PCA works here. And yeah, like this, what I'm doing, I'm just changing the numbers of principal components, and we can plot them on one axis or another and check at which configuration we have some better separation between different classes. And then, like a side effect of the dimension reduction done with PCI, we can extract also the original features loadings, which means what the fraction of variance is explained by the particular wavelength. So we're taking the principal component 1, for example, and evaluating which wavelength took the biggest loading, so the biggest variance, could explain the biggest variance within this principal component. So to get it integrated like in one curve, because you have this parameter set for these loadings set for every principal component. We can calculate it cumulatively, so just plus all the loadings from first 3, or in our case it could be also 10 components. And here we plot those importances or the loadings along the original spectrum. Here's an interesting information. So you can see that the most important parts of those spectra are the parts where the slope are the steepest. So you see that we're getting the highest loadings in the regions where the slope is going up, then we're getting drop in the loadings where the slope is going flat, and then again we're getting a peak where the slope is changing quickly again. And this image is actually very interesting and insightful. So if you imagine how would the derivative look of those spectra. So if you take the derivative of this spectral curve, you can imagine that it looks exactly like this. So here essentially we are mapping so we are telling the user who is analyzing the data that the most important thing for the distinguishing between the classes in our case is the place where the slope starts, ends, and what the degree, so what the scale, what the values within the slopes are. And this is exactly what would show you the function of the first derivative if you calculate from the spectrum. So it's kind of small hint. What else can be done with a radio spectrum to extract useful information? Especially it works for the visual spectrum, as for the spectrum in visual plus near infrared plus sphere range. And yeah, so then we can also evaluate this importance. So we can visualize and or print top 10 or 50 wavelength. You see that up to here we are just describing, up to here we're just describing one peak, smooth one. So, but this is the one which was described, and then we are also just marking top wavelength on the second one. So if you want to create some vegetation index, which would be super useful for extracting, like separating our calendula cultivars between each other, this formula probably should look like something 532 nanometers minus 687 nanometers divided by 532 plus 687, and probably you'll get some vegetation index which will look nice for your purpose. So that's how PCA can be used for the generation of vegetation indexes which were not used, which were not created, like existing before, custom made for your study. And the last step, I will show you how same data, not PCA one, but the original spectral data can be used for the training of the basic machine learning model, something similar to what I've shown you in the intro part. So the probably the simplest, one of the simplest methods, models you can use for the classification of such kind of data is Lasso regression. So this is kind of linear regression method with a so-called Lasso or L1, if I'm not mistaken. Yeah, I think it's L1 normalization, regularization, which allows you to evaluate multidimensional data. So if it would be just classical logistic regression without regularization, that would be performing much worse. But so this L1 or mixed L1 plus L2, so-called elastic net normalization, those should be quite suitable for the classification of our data. If we just take a look where this model comes from, so here it's at this part, logistic regression L1 and L2, and we were also evaluating it in our study. It showed okay results, though we have some models which were much more performant. But for some basic illustration, It would be sufficient here. So we separate our data to the numeric data X, to the labels in Y. Then we need to encode our labels into the numeric format because this model doesn't understand strings. So our original class names were named like L16, L4. So we are converting here them to the numerics. Then we're splitting our dataset to two random sets, train and test. So the ratio we can modify here. Currently we have 70% of data sent to the train one, so the data on which our dataset will be, our model will be trained, and 30% are left for the test. So we can verify with the data model, and we also click the stratify option. We set it to the to stratify by the encoded labels, and this function will be taking care of even splitting of our classes. For example, if we imagine that we have two classes in our dataset and we have ratio like 40% to 60%, this stratify method will take care that it will be splitted to test and train, and in which test and train it will be still 60% to 40%, so that we don't have by random split, we don't have our data distribution skewed. So then we are defining the Lasso object. So we are setting the Lasso regression. We are setting some basic parameters. One more thing which can be done here is that we can screen for best parameters for this regression. I'm absolutely not sure that those parameters are the best ones, but this is one more thing which probably goes for the next iteration of the workshop, if anyone will be interested. So the hyperparameters search, hyperparameters tuning, because every model can behave very much differently depending on which hyperparameters were selected during the initiation. And then we are training our models with our numeric data and with our labels. Then we are predicting, so validating our model on the test dataset. Then when converting our labels back to the string formats and printing results together with, so printing metrics together with plotting the confusion matrix. And here we go, so it should be quite quick, I believe. Okay, and we can see that the accuracy here is actually not of anything impressive. So we've got kind of, so 041 is rather weak. We've got some classes where we have the accuracy higher, so accuracy F1 score, so higher. We've got some classes where we have very low, so definitely not the best, results. And just one experiment I want to try here, because as I mentioned before, usually it's quite complicated for a model to, especially such a simple one, to comprehend all the vastness of spectral data. And very often it can be actually much more useful to use the data with, to use the data after the dimension reduction, just to prevent, data from the, just to help our simple model to handle generalization well, because dimension reduction is taking care of extracting most valuable data, and then extreme amount of dimensions doesn't overwhelm the model, and the model should not solve a lot of interdependent wavelengths, because as I mentioned, our spectral features are usually mutually strongly dependent. Each wavelength is not completely independent. We have smooth spectrum, so we need to handle a lot of mutually dependent data. And when we are applying dimension reduction, we can kind of encode those complicated interrelationships into the simpler principal components. And we have less of these principal components. So let's check one thing. What happens if we just instead of original raw spectral data frame, we will use the data frame after dimension reduction? What results we'll get? And we actually get something like 10% improvement of the accuracy right away. And it's only one of the steps which can be done, really. So nice. That's already something on the merge of usability. You see that the F-score, one score for some classes is actually quite okay, so 67% is good. And then, well, the teaser, what can be done next? And as I mentioned before, we are not doing much on the side of the machine learning preparation steps here. So the first thing which probably can be done is we can do a little bit more sophisticated analysis, including like PCA or different dimension reduction methods like LDA, PLSR, or like embedding like TSNA. So though this is rather useful for the visualization, I would not use it for any modeling. So then we can use techniques like smooth for data balancing, because as you've seen, we have quite different amount of data for every class, and for classifier it can be important. So we have the balanced amount of the classes, so we have enough representatives for every class. And then, so we can use different normalization techniques. So, as I suggested before, the standard normal variate or robust normal variate would be better candidates for our case, maybe not so illustrative, visually appealing. So then if we switch to what can be improved in the machine learning part, so definitely we can use different models. As you can see from the presentation, the intro part, models like support vector machines and models which includes dimension reduction plus some nonlinear models like random forest plus PCA, or yeah, multi-layered perceptron plus PCA can be quite efficient in this. And again, yeah, so returning back to the also the question of hyperparameter optimization, because even with the correctly selected models, you need to find the optimal hyperparameters. It's not so easy task. So you need to find the best mode in which your model operates, and then yeah, and parameters at which your model won't be overfitting, so you still can do quite reliable predictions in different conditions. Concerning that we work in the field conditions where the illumination is constantly jumping, that can be quite critical step to prevent overfitting. And then we can, of course, just provide simply better data by implementing into our pipeline interactive image annotation and therefore better segmentation. Because now, as you remember, we were doing almost everything manually. We were setting the thresholds for the segmentation manually. We can do it with the different tool for image segment annotation, then supply it for the segmentation model, and create model which will be much more robust and flexible for the segmentation itself. But everything of this probably can be again shown on the consequent workshops if any of you will have an interest. At the end of the mail where you found this link to this video, you can also find the Google Forms. And for any of you who would like, you can go there and select the topic for the consequent workshop if you want us to create it, if you want us to explain something for you. And well, when we evaluate it, you can suggest your topic, you can vote for the topics which are inside. And after we evaluate it, we can organize the second or third session if it would be necessary. This time probably solely online. And yeah, just demonstration what this changes, which I mentioned, can do to the same, absolutely same data. So what was done? So preprocessing was adjusted, data were annotated and segmented with the interactive annotation and machine learning for segmentation. Then it was applied to different model, dimension reduction and nonlinear model, in this case random forest plus PCA. Then we used also extensive hyperparameters tuning, and you can see that metrics for the data improved quite significantly. So we have a 1 score to have accuracy of Y score. It is very high, around 1, and the confusion matrix is showing absolutely different story than shown before. So here we barely can distinguish several classes from each other, and here we have very nice, very clean classification quality. So then also with additional visualization you can visualize important features not only from PCA, but here we actually using the projected loadings onto the features of the random forest. They were shown combined importance. So you see that this images have much